Alright, so in this video, I'm just going to give a brief overview of enzymes, and I'm going to give an example of an enzyme, and this video is going to be suitable for about GCSE level, or for someone who's maybe doing A level, and just wants a quick recap on the fundamentals of enzyme action. Okay, so we're going to start talking about enzymes specifically. Firstly, what is an enzyme? Well, an enzyme is simply a catalyst. Specifically, it's an organic catalyst that speeds up the rate of a chemical reaction within the body. Now, it's important to understand that a catalyst, an enzyme in this case, is going to be able to speed up the rate of a chemical reaction, but is not going to get used up, or is not going to be changed itself. So it will be able to continually catalyze a reaction over and over and over again until it runs out of things to react with. So they're not consumed, and they can continue to catalyze any reactants that they come into contact with throughout a reaction. Now, we're going to talk about reactants and products. In this case, the enzyme will work on the reactant. The reactant is the molecule that's going to be either broken down or in some cases joined together. Okay, now with a catalyst and with an enzyme, you only need small amounts of that enzyme in order to increase the rate of reaction. And it reduces the amount of energy that is required for the reaction to take place. Alright, so moving on. We're going to talk about hydrogen peroxide. Hydrogen peroxide, H2O2. Now this is a chemical that is not to be confused with water, which is H2O. This has got another oxygen molecule found within the compound, and it changes the properties significantly. For example, H2O, water, very, very healthy. We require it to live. We are majoritively made up of water. H2O2, however, even with just that one extra molecule, is extremely toxic to body cells. It's extremely toxic to cells, and therefore it has to be removed from the system as quickly as possible so that it doesn't damage any of your cells. Now, hydrogen peroxide is produced as a waste product within the body. So when you're metabolizing, you're undergoing chemical reactions within your body, you are producing hydrogen peroxide as a waste product, and it has to be removed before it can damage the cells. Now, it does that using an enzyme called catalase. You can tell catalase is an enzyme because it ends with the phrase ase. Lipase, protease, carbohydrates, these are all enzymes. In this case, the one that breaks down hydrogen peroxide is catalase. Now, catalase can break down hydrogen peroxide, which is extremely toxic, into water and oxygen. Two very safe and essential chemicals that the body requires. Okay, so in this case, one catalase molecule is able to convert in the human body roughly 5 million hydrogen peroxide molecules per second. It has an extremely high turnover, and because of that, not much catalase is actually required in order to metabolize a significant amount of hydrogen peroxide. Now, catalase is made up of 4 polypeptide chains. 4 polypeptide chains, essentially meaning that catalase, the enzyme, is a protein. And as a protein, it has a 3-dimensional structure. And this is important to understand this part, in that this structure is highly specific. It's highly specific to the reactants that the enzyme is going to interact with. So the fundamental action of an enzyme comes down to the most simple theory, which is a lock and key hypothesis. It's the very basics of enzyme action. Obviously, they actually are a little bit more complicated than that, but we're going to discuss the lock and key hypothesis in this video. Now, in the case of hydrogen peroxide, okay, the diagram we've got here shows the enzyme, catalase, is the red molecule, and the yellow molecule is the substrate. Now, in the diagram it says food particle, but in this case we're going to be talking about hydrogen peroxide. Hydrogen peroxide is the substrate, and catalase is the enzyme. Okay, so the red one's the enzyme, the yellow one is hydrogen peroxide. Now, the enzyme catalase has got an almost very specific active site. The active site is a part of the enzyme that the substrate binds with. Okay, so the substrate is very specific to the shape of the active site within the enzyme. You can see on the diagram that the substrate fits nicely into the enzyme's active site. Okay, now technically speaking, the active site is a little bit more flexible than this would have you believe. It does actually mold to the shape of the substrate, but again, it's very much an exclusive thing. This enzyme is specifically for the breaking down of hydrogen peroxide, the metabolization of hydrogen peroxide into oxygen and water. So what happens is the enzyme will move along, and because chemical reactions have got a certain amount of energy in them, the substrate and the enzyme will all be moving around within the cell. So the enzyme is going to bump into the substrate, and when it does, it will take the substrate into its active site, as you can see in the beginning of this diagram. Now once the substrate is in the active site, you get a combined enzyme and substrate. Now this is called an enzyme-substrate complex. Immediately after that happens, the catalase, the enzyme, will hydrolyze, that is, break down the hydrogen peroxide into oxygen and water. These are your products. So if we follow the diagram, enzyme binds to the substrate, so catalase binds to hydrogen peroxide. It undergoes hydrolysis, so it breaks down the hydrogen peroxide into oxygen and water, which we call the products. These are three very important words to remember: substrate, enzyme-substrate complex, and product. And these are the fundamental words you're going to use in all enzymatic reactions. Okay, now this happens extremely quickly, and the enzyme is left unchanged. Whereas the substrate has been broken up into two component parts, oxygen and water, the enzyme is unchanged and can then move along and break down another hydrogen peroxide molecule. Alright, so the substrate is hydrogen peroxide. It's what's being changed. Starts off as H2O2 and becomes water and oxygen. Alright, now, catalase is worth remembering. The catalase is found in almost all living things. Okay, it's found in pretty much every single living organism on the planet. Okay, plants, animals, bacteria, many different organisms. Now, catalase speeds up the breakdown of hydrogen peroxide so that they are not toxic, so that they are unable to harm the cells with their toxicity. Right, so heat. Inside the body, you've got a base temperature of roughly 37 degrees centigrade, at 37.7 degrees centigrade. This is the optimum temperature for enzyme activity. It gives the enzymes plenty of energy, which means that they're able to move around and they're able to come into contact with the substrate fairly quickly. Okay, now, if you get a fever, or if you are too hot, or if there's too much heat provided to the reaction, then the enzymes can denature. Denaturation is a process by which the 3D structure of the enzyme, the actual proteins and polypeptides themselves, become deformed. If they become deformed, the active site in the enzyme will not fit the substrate. If the substrate does not fit into the active site, that substrate cannot be broken down, it cannot be hydrolyzed, and the enzyme will not be able to increase the rate of that chemical reaction. Now when you go the other way and you get too little temperature, something that's too cold, enzymes will be given less energy. If they've got less energy, they'll be moving slower. Now fundamental reaction theory suggests that reactions occur when two molecules collide. And in order to collide, they've got to be moving. Now the slower they're moving, the less collisions are going to occur. So the enzyme is going to come into contact with the substrate less frequently. This means that the rate of reaction is going to be slowed down. So if you become too cold, your rate of enzymatic reaction becomes significantly lower. And that can be a problem, specifically with hydrogen peroxide. As hydrogen peroxide is toxic to the cells, you don't want it staying in the cells for too long. Right, now, there is another chemical, another catalyst, okay, that is able to break down hydrogen peroxide into oxygen and water, and that is manganese dioxide, MnO2. The question is that therefore an enzyme, seeing as it does essentially the same job as catalase? Well, the answer is no. Manganese dioxide is a man-made chemical. It is an inorganic catalyst. While it is capable of breaking down hydrogen peroxide into water and oxygen, it is not organic, therefore it is not an enzyme. An enzyme specifically is an organic molecule, a protein, polypeptide, that is capable of breaking down hydrogen peroxide into water and oxygen. Manganese dioxide is inorganic, it's man-made. They both do the same job, but one of them is an enzyme, catalase, and the other is not. I hope this video has helped you gain a fundamental understanding of basic enzyme theory. If I've made any errors, please leave it in the comments below.